All right, so welcome back, everyone. I'm Javi, and I'm going to be moderating 6 great talks that we have in this session. These are 8 to 10 minute talks, and then we'll have some minutes to ask questions if you like. So our first speaker is Olaf Canos, a PhD student at the Barcelona Supercomputing Center and the IRV. And her talk is going to be about metagenomics in the field of cancer. So I play now the talk.
Hi, everybody. I'm Olfat from Comparative Genomics Lab, led by Professor Toni Gabaldon at the Barcelona Supercomputing Center and Institute for Research in Biomedicine of Barcelona. For me, it's a pleasure today to introduce some of the work that I'm doing as a PhD student. And specifically, today I will talk about microbiome profiling from fecal immunochemical test samples as a source of biomarkers for colorectal cancer screening. So the overall goal of my PhD thesis is to develop and implement computational tools in metagenomics for the finding and functional interpretations of biomarkers with a diagnostic value. In this case, today I would like to talk to you about a project in relation to colorectal cancer. So I'll give you a brief introduction to contextualize you, and then I'll show you some of the results that we have been obtaining. So let's start. Colorectal cancer is the third most common cancer and the second leading cause of cancer deaths worldwide, being its incidence a healthcare challenge. It has different molecular phenotypes and strong resistance to therapies. Therefore, there is a need to find biomarkers and develop new tools for early diagnosis. It is known that this malignant disease involves both genetic and environmental factors, and that the lifestyle and the diet are playing a significant role in the development of the disease, likely through an alteration of the gut microbiota. So, what is the gut microbiota? The gut microbiota, also called as gut microbiome, is the set of microorganisms that are inhabiting the gastrointestinal tract, being the colon the most heavily colonized part, containing approximately 70% of our human microbiome. It is known that alterations in the gut microbiome, what we call dysbiosis, can influence tumorigenesis. So our knowledge of the microbiome has been expanded during the last years due to the advent of gene amplicon techniques and metagenomics. So the most widely used techniques within each of the groups are the 16S RNA sequencing and the whole shotgun sequencing. Colorectal cancer diagnosis is challenging. So normally starts with some symptoms in the patients that are ending up with a final diagnosis by the gold standard technique, which is the colonoscopy. However, as you may know, colonoscopy is invasive, time-consuming, and expensive. So a lot of efforts are needed in order to enhance the accuracy of pre-colonoscopy tests to derive the cases that should be subjected to colonoscopy or not. So here in Catalonia, as implemented in other regions, there is this fecal immunochemical test that signals if there is hidden blood in the stool. If it's positive, then a colonoscopy is carried out. However, the false positive rate is high. In this context appeared the CREEPREF project, in which the main goal is to derive a risk model by combining the FIT test result with other information that we can obtain from the same tube, such as the microbiome profiles that we were studying in the group. So this is the overall methodology that we have been applying. So from these fecal immunochemical test samples, we obtained the 16S RNA sequencing reads. We applied a pipeline, obtaining what we call the amplicon sequence variant table, which is a table that allows us to find which taxa we have and in which relative abundances for each of the samples. So combining this table with the metadata, we could perform different analyses that I'm going to show you now. So let's talk about which is the data that we have. So around 3,000 samples from the Catalan screening, and not only considering colorectal cancer and negative samples after the colonoscopy, but also different lesions that are relevant in the development of the disease. We also classified our samples according if they are clinically relevant or non-clinically relevant. So we started with a characterization of which taxa we could assign. And well, here we have a representation of the 10 top most abundant taxa. And this allows us to verify and corroborate the use of fecal immunochemical test samples to study the gut microbiome, because by using the same tube, we could make the hemoglobin detection and the segment of taxonomy, and this composition was similar than the one that is observed when doing it with stool samples. So we have no the need of collect the costly and complex stool samples. We also studied the beta diversity of our samples, so the diversity between samples, so how different is one sample to another one in terms of the overall composition. And as you can observe in this multidimensional plot in which we represented the Edgestone distance, there is no clear clustering of our samples according to the diagnosis. However, applying an Adonis test, we detected a significant effect of the diagnosis combined with sex and age. So we were interested to find which are the main drivers of this effect of the diagnosis. So we made a differential abundant analysis by applying this linear model, and we detected 34 taxa at species level as differentially abundant according to the diagnosis. By applying a multiple comparison, we detected that the most distinct group across this path from negative to colorectal cancer was the colorectal cancer. That's why we applied the same linear model, but so considering colorectal cancer versus the others. And we did the same by comparing clinically relevant versus non-clinically relevant. We also made a functional prediction. So from our taxonomy profiles, we predicted, we inferred some orthologous groups, and we applied the differential abundant analysis, obtaining differentially abundant orthologous groups. And from here, we extracted some pathways, and we defined some enriched pathways according to the diagnosis. So here we have the ones that we found. I'm not going to enter in detail because I have not a lot of time, but with a text mining tool, we also included the number of articles in PubMed that are linking these pathways with colorectal cancer. So taking profit that we observed distinct features, so distinct compositions of in terms of the taxa in our samples according to the diagnosis, we wanted to derive a classifier to detect which samples can benefit more from colonoscopy intervention. And we focused our attention to have a good recall, because false negatives are of more medical concern than false positives. So to make the long story short, we have a strategy of a two-phase classification. In a first classification, we are classifying colorectal cancer versus the non-colorectal cancer, and then the ones that are not classified as colorectal cancer are subjected to a second classification, which is clinically relevant versus non-clinically relevant. So we applied a feature selection based on our differentially abundant results. And when including more taxa, we are having a better area under the curve and specificity. But as I said, we are interested to magnify, so to have better recall and sensitivity. So when restricting only to 4 taxa panel for each of the phases, we obtain better recall and sensitivity for colorectal cancer. So the specificity, so the false positive reduction that we obtained was around 21%. These are results of an average because we applied the strategy 100 times in order to avoid likely splits. So in the context of the current system in Catalonia of screening, if we simulate with our numbers, considering this system, and we consider 100 participants, we are saving 556 colonoscopy while magnifying the inclusion of clinically relevant cases. So let me finish this talk by giving you some take-home messages. Our results corroborate the validity of feed samples for microbiome analysis, avoiding the complex and costly collection and processing of separate stool samples. that colorectal cancer is the group along the adenoma-carcinoma path that have the most distinct microbiome profile, that small changes matter, that we reconstructed changes in the composition and metabolic patterns that are associated to clinically relevant features hinting to potential microbial roles in the origin of colorectal cancer, and that we developed a two-phase classifier that achieves close to 100 sensitivity for colorectal cancer while significantly reducing the current false positive rate. Let me thank Tony Gabaldon to give me the chance to join the group and to perform this exciting research. Also Jesse and Esther for all their help, and to all the comparative genomics lab, and of course to you for your attention.
All right, Olfa, thank you very much. Very clear explanation. So let me check if we have questions. So I invite everyone to make questions to Olfat, and I'll start with one in the meantime. So I was, well, you said that you focused on the recall metric to be more aware of false negatives. So, and then I was wondering, in a clinical setting, how would you deal with the false positive samples? I mean, can you increase also that metric, the precision metric?
Yeah, well, in this regard, in this classifier, we are only including so the taxa that I commented, but also the sex, the age, and the fit value. So maybe including more clinical data will allow us to increase decrease the false positive rate. Mm-hmm. Because yeah, it's a problem for the current system, for the healthcare system.
Mm-hmm. And I missed about the sample size. I mean, the different groups, they were well balanced?
No, they are not balanced, but in order to, well, to take in consideration this, what we did was to give more penalty to the minor class. So changing the weights in the models when training, in order to consider that there is this unbalanced classes. But yeah, this is a problem, and I think that it's a problem in all the yeah when trying to define these models.
Very good. And do you see? Last question I have is like, do you see in the future your strategy to be applied in different types of cancer and different type of sample types? Perhaps it's a very difficult question to translate that.
Well, I think that we should explore more. I mean, this is only the beginning, but we have tight collaborations with hospitals and clinicians, and they are super enthusiastic with this type of analysis. And I think that in the future, maybe it can be a reality. So yeah.
Well, we'll have So a question just popped up from Guillermo. Let me show it here. So it says, thank you very much for your presentation. And out of curiosity, is there any percentage of the sequences in your samples that could not be identified?
So when I think that he's referring on the taxa that we could not classify it, and yes, so with 16S, one of the problems is that at the species level, in a lot of cases, we cannot arrive. So we can only classify at the genus level. And well, in order to, well, we are planifying a project by using whole shotgun sequencing in order to tackle this problem. But yes, there are some sequences that we could not classify them.
All right. Thank you very much for your answer and for your question, Guillermo. So, and I think we will move to the next talk. So, but very interesting, really.
Thank you
very much.
Thank you.
All right. So, okay, let me find it here. Okay, so yeah, thank you very much for sending us your work. Edurne is our next speaker, Edurne Urrutia, and she's doing, I mean, she will present the work that she did during her final degree project at the Príncipe Felipe in Valencia. So I will just play the video and let's enjoy.
I'm Edurne Urrutia, and today's presentation is based on my final degree project, a systematic review and meta-analysis of transcriptomic studies unveil sex differences in rheumatoid arthritis. I know it's not the catchiest title, but it will get better. First, I talk briefly about the disease, just to put it in context. I continue with the aim of the study, how we achieved it and the main results we obtained, and I'll finish with some conclusions. Rheumatoid arthritis is a chronic autoimmune disease. It's characterized by joint inflammation, bone destruction, and the development of autoantibodies. The antibodies get attached to the lining of the joints and trigger the attack of the immune system cells, causing inflammation, pain, and bone erosion. Although it has a strong genetic component, it's a complex disease with many associated risk factors, including sex. In fact, rheumatoid arthritis, like most autoimmune diseases, shows important differences between men and women. It's 3 times more frequent in women than in men. Men, instead, have a later onset and are more likely to achieve remission. Sex hormones are known to be involved, but the differences in the molecular mechanisms are not fully understood. That's why, despite these differences, diagnosis and treatment are surprisingly the same for men and women. To address this issue, the aim of the study was to shed some light on the molecular mechanisms underlying sex differences in rheumatoid arthritis. Further knowledge may allow to find biomarkers that could foster personalized medicine. The goal was achieved by first conducting a systematic review of rheumatoid arthritis studies, second, assessing the selected studies individually, and third, performing a gene meta-analysis followed by a functional arrangement and thus obtaining information about the molecular mechanisms involved. Here we review the 3 steps in more depth. All of them were carried out using R programming language and libraries. The first step was the systematic review of rheumatoid arthritis studies. The studies had to include healthy individuals and patients with gene expression data from either microarrays or RNA-seq platforms. For this, we checked the public repositories Gene Expression Omnibus, ArrayExpress, and ADEX. Then we selected them according to the PRISMA statement. The selection criteria were the use of human samples with at least 3 individuals in each experimental group and available sex data. Those based on cell cultures or undergoing ex vivo treatments were excluded. Second, we assessed the selected studies individually, first processing their data and then performing a differential expression analysis. Data processing included downloading the data, normalizing the raw count matrices from the RNA-seq studies, and screening the samples. An exploratory analysis was conducted to review the range of values and the detection of outliers. We also checked the sample distribution in the experimental groups and the presence of batch effects. Then we classified the samples into 4 experimental groups: control women and control men, and ill women and ill men. Finally, we also replaced the probe and gene identifiers by entry identifiers. As a result, we obtained consistent datasets among studies. The next step was the differential expression analysis. It involved 3 contrasts: in women versus healthy women, in men versus healthy men, and the differences between these 2. The latter reveals the differentially expressed genes between ill women and men. After the individual assessment, we performed a gene meta-analysis. We chose a random effects model because it takes into account the differences in the studies combined in the meta-analysis. This step results in a long list of genes ranged according to their differential expression, but extracting meaningful information from the list is quite challenging. So we additionally performed a functional arrangement from the overall results of the meta-analysis. Thus, we detected a set of genes that were coordinately overexpressed or underexpressed and involved in specific functions, according to the biological processes from the gene ontology or GEG pathways. These results were synthesized using Gauss-Lim terms and the Rovigo tool. I will now describe some of the results obtained throughout the process. This slide displays a prisma diagram. It shows the flow of the studies during the systematic review and the number of remaining datasets in each phase. All in all, we identified 200 candidates, which led to 6 useful studies, 2 RNA-seq and 4 microarrays. The main reasons for exclusion were the lack of control groups and sex. The next step, the individual assessment, ensures that the studies are comparable. As a result of this approach, we finally retrieved 5 datasets with 570 samples from the GEO repository. Then we performed a gene-by-gene meta-analysis, which identified 10 genes, 90 geobiological processes, and 11 key pathways as significantly sex-dependent. As I previously said, the results were summarized in 2 complementary ways, the Rovigo tool and Gauss-Lim terms. This pyramid shows the number of co-terms associated to each Gauss-Lim term. Terms overrepresented in male men are in red, and those in women are in blue. These figures suggest a higher activity of the immune system and a greater presence of energy precursors in female patients. It's well known that women show stronger immune responses than men due to sex hormones. This activation of the immune system requires a large amount of energy, which could explain the greater presence of energy precursors. Male patients instead showed an overrepresentation of terms related to protein biosynthesis. Studies suggest its relationship with a high degree of inflammation. In women, this could be partially counteracted by the anti-inflammatory action of estrogens, thus explaining the sex difference. Although more research is needed, differential expression of inflammation between the sexes could be a therapeutic target for rheumatoid arthritis. This last figure was obtained using the Rovigo tool, and it's based on semantic similarity. In addition to the immune activity in women and protein synthesis in men, the graph highlights the negative regulation of osteoblasts in men. The osteoblast-mediated decrease in bone formation is, together with erosion, the cause of bone loss. This agrees with bone loss being one of the symptoms of arthritis and has been reported more frequently in men. Hence, the negative regulation of ossification could be an underlying mechanism of sex-based differences in rheumatoid arthritis. So let's finish with some conclusions. The systematic review shows that sex is still an overlooked variable in biomedical research. On the one hand, women show a higher response of the immune system and a greater generation of energy precursor metabolites. The latter may be related to the energy expenditure required for the immune response. On the other hand, protein biosynthesis and negative regulation of osteoblasts are noteworthy in men. These mechanisms could explain some of the differences between the sexes. Thus, our findings could lead to the discovery of new therapeutic targets and be the basis for future research devoted to sex-specific precision medicine in rheumatoid arthritis. Thank you so much for your time. See you soon.
All right, Edurne, congratulations for your work. Very nice talk. So let me check. There are questions already here. Okay, no by the moment, but I'll make some of them to start. Well, first of all, this is a very beautiful example to understand sex differences, and it's very nice to see this kind of topics covered here after the Bioinfo for Women talk that we have in the morning. And I was first, I didn't get the gene expression data that you used. What kind of samples do they come from?
Okay, they were, well, first of all, thank you for inviting me here.
I tried to do my best answering because I'm a rookie. But yes, there were 4 studies from peripheral blood, and there was a last one with synovial tissue samples.
Sorry, synovial.
Yeah.
Okay. Thank you. And I very easy question that I am sure that you know is like, what type of normalization did you apply in the counts? That
TMM.
TMM normalization.
Yeah.
Okay, because I'm not pretty confident with that, and I wasn't sure. And is there any other?
Yeah, I didn't mention it. But,
is there any other methodology for normalization that you tested, or?
Well, no, in this study, no, because my team has been performing this type of meta-analysis for a long time. But yeah, if you want, I can take your email and write you with the details because I don't have them on the top of my head.
All right, thank you. I'll take note of that. So, all right. You guys have any question there in the audience to Lourdes? In the meantime, why do you think that male did not show any enrichment in the immune system?
They did show an enrichment in the immune system in specific genes, but it's, well, are you referring to the last pyramid, no?
Yeah, exactly. Yeah, it was a blue bar at the top that it was only enriched for women, I think.
Yeah.
This is really long bar, and there is 0 for male. So I mean, it's a strong effect, right?
Yeah, really strong. It's related to the presentation of antigens, but I can't recall why there are no functions in men, because in the differential expression analysis there were some genes related to the immune system, but yeah, in the functional arrangement they didn't appear, and I can't recall why.
There might be a reason then. Yeah, perhaps this work has been done a long time ago or a bit of time ago, so it doesn't matter. It's just a question. All right, and well, no questions on the panel by the moment, so we can spend 1 more minute to wait and see if there are more questions. And another question that I could make is about, as I asked Olfat before, about the number of samples in each group. I mean, do you think if it was well balanced, or?
Intra-study, they were well balanced, but not inter-study, because there were very big studies and some of them were quite small. But yeah, intra-study, they were quite balanced.
Okay. All right. So, well, there are no questions by the moment, so I'm gonna pass to the next speaker. But thank you very much for your work and congratulations.
Thank you so much.
Take care. Bye. Okay. Very good. So, right, it's now time to listen to Hector Rodriguez from University La Laguna and Hospital La Candelaria in Tenerife. And we'll see a talk about metagenomics and nanopore technology. So I'm going to play now the video. So the stage is going to be all yours, Hector.
My name is Hector Rodriguez, and I am a PhD student at Hospital Universitario La Candelaria and Universidad de La Laguna. I'm very pleased to be here at this 7th Student Symposium on Bioinformatics from the RSG-Spain. In this talk, I want to present you our new development, Nanotax, a real-time pipeline for taxonomic and diversity analysis of Nanopore 16S RNA amplicon sequencing data. Throughout this presentation, I'm going to introduce 16S rRNA amplicon sequencing on Nanopore devices and bioinformatic analysis software methods for microbial sequence classification. I will describe the Nanotax pipeline and make some comments about the implementation details. For the second half of the talk, I will describe the results that we have obtained with human microbiome samples and the Nanotax workflow, and I will finish with some conclusions and future perspectives on nanopore sequencing and the real-time workflows and applications. Nanopore long-read sequencing have enhanced the study of microbial communities, providing more versatile and efficient approaches that bring sequencing assays to small labs and unspecialized facilities. The technology itself provides rapid protocols, multiplexed libraries which allow up to 96 samples in the same flow cell, and real-time analysis of sequence data. Regarding technical aspects for analysis, the Nanopore devices allows to sequence the full-length 16S rRNA, which instantly translate in better taxonomic resolution and reduce the bias introduced by the choice of 16S subregions, as happened in short-read assays. From a bioinformatics side of view, the classic approach for taxonomic analysis of microbial sequence data relies on the classification of individual breed against sequence databases or the alignment against a set of genomes of interest. The golden standard for sequence classification is the well-known tool BLAST. Despite the advances in efficiency and speed of classification methods, the BLAST tool still provides interesting features like useful metrics for results interpretation. On the other hand, we have rapid classification tools and aligners. These approaches are also based on the search of a query sequence in a database, but feature big enhancements on search speed and index storage. These rapid methods have leveraged the analysis of huge sequencing datasets as the ones we get at next-generation sequencing workflows, although presenting more challenges at the results interpretation. The Nanotax Pipeline features a complete taxonomic and diversity analysis workflow, which include not only classification, but downstream analysis and visualization. Pipeline input consists of FASTQ files directly extracted from finished or ongoing sequencing experiments. In this real-time mode, the pipeline runs endlessly and is fed by the newly generated FASTQ files. Reads pass through a quality control step and are classified using the software of choice from Kraken 2, Centrifuge, and BLAST. To enable comparisons between methods, the output then pass through a downstream process, which aggregate the new data into the results and generate the final reports. Apart from classic text-based output updated in real time, we provide a Python Dash web application to easily inspect Nanotax results in real time. This application doesn't depend on the Nanotax pipeline and can be installed separately. Results for finished and ongoing pipeline executions are presented in an interactive dashboard that includes sample composition and diversity information at the taxonomic level of choice. For ease of use, a conda environment and Docker container is also available for running the web application. To assess the usefulness of Nanotax in the context of human microbiome samples, we have reanalyzed a set of 31 tracheal aspirates from intensive care unit patients. In a different study, we observed that a reduction in the genus-level bacterial lung diversity is associated with intensive care unit mortality. Samples were analyzed in time windows simulating different stages of a sequencing experiment. We observed the same reduction in Shannon diversity index in deceased patients as early as at 2 hours of the sequencing experiment starts, which roughly correspond to 5,000 reads, depending on the sample yield. Additionally, we have assessed the Shannon diversity index as a predictive value for non-pulmonary sepsis patients' outcome. These results were essentially equivalent to those obtained collecting reads up to 24 to 48 hours that roughly correspond to 100,000 reads, and suggest the importance of bringing sequencing to critical time environments such as intensive care units. The Nanotax pipeline can be used to benchmark different classifiers and databases. Additional parameterization could enable the analysis of different amplicon sets or shotgun metagenomics data. Complexity reduction in pipeline and dependencies installation are key for distribute your pipelines and methods. We anticipate that pipeline engines will be broadly used in the future as they provide a simple framework for writing reproducible and scalable software. Nextflow versatility have enabled, for example, the implementation of a real-time analysis workflow like Nanotax. Despite the real-time capability being present since launch, applications are relatively scarce. We anticipate that advances in quality and the cost of the technology will bring sequencing to more and more labs, resulting in more bioinformatic advances and applications in critical time environments like the clinic. To finish this talk, I would like to acknowledge my peer Laura Xufreda for her support during the development of Nanotax, my mentor Carlos Flores for his advice and support, and all the people in my group that inspire me daily. Also, thank you to all the people in the organization for this event and this opportunity to share our work. Now you can feel free to ask any questions about this presentation and Nanotax. Thank you.
So Hector, congratulations for your talk. Thank you very much for participating.
Thank you.
So we'll wait for questions. So please, guys, come on, make some questions over the panel. And I took note I took some notes, and I have some easy questions. The first thing is I also use BLAST sometimes just for one sequence or something very isolated. But how do you run BLAST? I mean, is it something more automatic? Do you use an API or something?
Yes, so for BLAST, we are essentially using the command line tool that I think is okay. It is just okay because it comes with some problems, you know, the databases, then you have to figure out what is every parameter doing in your analysis. And this for me was very hard to do because I also came from the web-based BLAST. And, you know, with command line interface, things are pretty different. But yes, it is just the BLAST-N tool that you download from Conda, and that's it. The BLAST process is just a normal BLAST execution.
All right. I'll give a go next time and try to do it in the command line as well. And also about your methods, I had understood that, for instance, Kraken needs a lot of RAM memory. Is it that true, or?
Yes. So the main advantage for using Kraken back in the days, back in the days 4 years ago, it was that Kraken was, it can make very quickly classifications, not as good as BLAST, but it was a very high speed improvement. But then they released the Kraken 2 version that essentially does the same, but the second version comes with these memory improvements that I think that makes possible to to run Kraken on a laptop, for instance.
All right. So
yes,
you need just 8 gigas or
yes. The mini Kraken database is about 8 gigs, and yes, it does not cover the same genomes as NCBI RepSeq, but it's pretty complete. It's the mini Kraken database. You can download some database that are around 16 gigs. Because I have in mind that Nanotax would be used not only on big workstations or computing facilities, but normal laptops as well.
That's super nice. And now that you mentioned Nanotax, you mentioned that it is written in Nextflow language. So do you have in mind to publish this in the NF core or any repositories for a bioinformatics pipeline so that everyone can use it?
Yeah, so actually I use the NF core blueprint because I think it's very straightforward. And yeah, it would be nice to publish it on the NF core suite. This is not the first pipeline that we built using Nextflow. We built NanoClast, but I think that was a more a specific method, and that was not a pipeline itself, but it was a method. But this time with Nanotax, I think that I could benefit from getting support from NF core people who are nice. I think that pipeline engines and frameworks and libraries such as Conda or Docker are nice, mostly because of the community. You have a problem, and then this guy came with a solution, and yes, that's really nice.
That's so cool. So good. Well, there are no questions by the moment. and we have some more minutes. So I was also thinking about the use of nanopore technology, which is kind of cool and easy and fast, as I have understood. So what nanopore configuration did you use? I mean, the type of machine, you mentioned it there, but what is the data yield that it produces for your, how many samples you can run at once or things like that?
Okay, so for the kind of assay that we are working on, these are for full length 16S RNA reads, and you can. It also depends on the sample quality. You know that you have some sample and analytical issues when you are doing metagenomics, but we are running 12 samples per flow cell on a MinION.
Okay.
But I've read that you can run even 96 samples. But what concerns me about that is the coverage that you get for every sample. We are getting, well, our goal at first was to reach the 100,000 reads per sample. I think that with 100,000 you can do something. But you could found some samples that have a very poor yield. There's no solution for that. You can reuse your flow cell, rerun again your sample, but you will find some samples that you can have 40,000 reads, and you know that's not enough if you are aiming to use these reads for a study. You know, maybe for a rapid analysis, benchmarking is okay, but yeah.
All right, so I'm following the question about nanopore. We have a question here in the audience, which from Miguel Angel. So it says, how much better is the microbiome characterization with nanopore as compared with other long-read technologies?
Okay, so I would say that the best thing of using nanopore for 16S is the portability and the efficiency and also the increase on read length. But the best thing for microbiome characterization and nanopore is to use a hybrid approach, using, for example, Illumina short reads and nanopore long reads, both on shotgun assays, and that finally makes the difference. I mean, we could be discussing if Illumina or nanopore, I mean, short read or long read are better for shotgun metagenomics. I'm not into that discussion. But definitely, if you combine both technologies in terms of assembly workflows, at least, I think that you can make the difference. And this is for me, it's the beginning of the revolution of microbiome studies at a good resolution.
So Pacific Bioscience is not a solution?
I've not tried Pacific Biosciences yet. But I hope that people do well, because that means that we can have that Illumina and Nanopore also want to do it okay and want to fight for the best methodology, and we can benefit from that.
Very nice. Well, thank you again for all your answers and the question from the audience and your work. Congratulations. We're going to move to the next speaker. All right. So our next speaker is Miguel Joshikora. So we have he's doing a PhD at the Barcelona Supercomputing Center, and he's gonna talk about the structural variation in your species of interest. So welcome, and I'm gonna play now your video on the stage.
My name is Miguel Joshikora from the Comparative Genomics Lab led by Toni Gavaldor, which is in the Barcelona Supercomputing Center and the Institute for Research in Biomedicine. And I'd really like to thank the organizers for giving me this opportunity to talk here about structural variants and how to detect them in your species of interest. And just for a brief introduction, I'd like to say that genetic variation is sort of pretty important in many fields. For example, some people are interested in how different variants give a higher risk of having some human disease, or how they can be related to antibiotic resistance. For example, in my PhD project, I am trying to understand how fungal pathogens become resistant to drugs through these genetic variants. They are also important for natural diversity. So, for example, these are the drawings of Darwin in his famous trip to the Galapagos about different bird species, where he was trying to understand the evolution of them. And of course, we all have heard about the SARS-CoV-2 variants. And actually, all these things have one thing in common, which is studying genomes and trying to find variants. And if I say variants, you will probably think about these guys, which are single nucleotide polymorphisms. These are these types of mutations where you have a single position in the genome that is changing. And of course, when we sequence these genomes with short reads, which is what we do most of the time, we can, it's easy to align these reads to a reference genome and identify these variants. So most of the studies about genetic variation have focused on these types of simple SNPs. But today I'm going to talk about another type of variants which have been overlooked in many studies. And these are these structural variants, which are changes in the genome that are large. They involve, for example, the deletions or duplications of large parts, which are usually larger than the read length. So from a technical perspective, they are really difficult to find. So they have been really understudied. But still, we know that they can be important. So, for example, they can influence different diseases. In this paper here, they show how structural variants can be the major source of inherited Parkinson's disease. And we also have a lot of relationship between genetic rearrangements and cancer. And on top of that, there are some studies showing the relationship between some duplications and drug resistance in fungal pathogens, which is actually my PhD project, which is what my PhD project is about. But in general, these kinds of variants are really less studied, and it's because of this difficult detection, which I was saying before. And still, I have to say that this detection is possible. So let's say that it's partially possible, although it's sometimes difficult. So there are some recent algorithms that take some short read sequencing data set, and they can identify structural variants. So in this case, for example, they could say this region has been duplicated in this gene of interest. The thing is that these tools are usually only tested on some model species, mostly human. So it's not really clear if you are working with a new species, it's not really clear how you should choose the parameters to run these programs. And it's also unclear what the accuracy of these tools is, because the accuracy sometimes is not very high. So, and this, for us, these were two very important questions, since we were studying the genomes of these fungal pathogens where nobody has used these kinds of tools before. So in order to make these tools available for us, we developed this pipeline, which we call personalized structural variation detection, which, like the short is Persuade. It takes short reads and a reference genome. It maps the reads and it uses several software to identify the structural variants. And of course, we don't really know how to run these programs, and we also don't know how to filter the outputs of them. So the Persuade automatically, it runs simulations to, it simulates structural variants on the reference genome, and then it finds the parameters that work best on these simulations. So it uses these parameters that are sort of optimized for the input on the actual data. And these simulations have similar properties to the input. So they have similar coverage or similar read lengths, so that they are really adapted to each of the inputs. And with these, this means that we can use this pipeline to do this variant detection for any species. We get a report of the accuracy, which means that we can know how much we can trust the results in our species. And more importantly, we can do structural variant detection with a single bash command that goes from the reads to the variants that are filtered and, in theory, called in the most accurate way possible. So we developed this pipeline, and we tested it on simulations and also in real data sets. Today, I'm just going to talk about the simulations, but we can talk more about it if you are interested. So we actually looked for data in these 6 species. These are 6 eukaryotes that cover large part of eukaryotic diversity. Here we have 3 fungal pathogens, 1 plant, and 2 animals, and we gathered 3 short read samples for each of them. We had these 2 questions. The first is, what is the calling accuracy? How well does Persuade work? And the other is, how do the parameters that are fit for one sample perform on the others? And you'll see how this is important for understanding how important these parameter optimization is. And when I say accuracy, we actually mean precision and recall, and also we mean the F value, which is like this harmonic mean between precision and recall. And what Persuade tries to do is to maximize this value. And the interesting thing about this value is that it is only high if both precision and recall are high. So back to the questions that we had, what's the calling accuracy of Persuade? What I'm showing you here is data for Candida glabrata, where we have the calling accuracy of different types of structural variants. So we have deletions, tandem duplications, inversions, insertions, translocations, and also the average of all of them. And here we see what's the accuracy when either using optimized parameters or the default ones of Persuade. And we can see how this optimization increases a lot the accuracy, almost from 0 to 1 in some cases. This was for Candida glabrata here, you see, and this is for each of the species, these 6 species that we tested Persuade on. And what we can see is like something similar. Persuade is important so this parameter optimization is important to have the maximum accuracy. And of course, this depends on the type of structural variant and also on the species. So each of these species has a different behavior, meaning that there's probably no universal rule. So there is not, these algorithms do not either work well or bad. It will depend on the genome you use them on. And the second question that we wanted to solve was whether the parameters that work well for one species, whether they work well on the others, and this will give us information about how important is it to optimize the parameters for each of the samples that you want to work on. So as I said, we run Persuade on all these different samples and different species, and this yielded different parameters on it for each of the samples. So we call these training parameters. We actually run these training parameters on the different samples, and here I'm showing the results of the F value of these runs. So we can see it's a bit complex, but something that is pretty obvious is that the parameters that work, for example, in Candida glabrata samples, they always work in the other Candida glabrata samples, but not necessarily on other species. And this is like general for the other species. So it seems that in general, each species has a unique set of best parameters. And this is pretty much everything. I hope that you remember that structural variants can be important and that we have developed this Persuade pipeline that can be useful for finding them in your species of interest. And also that each species has a unique set of parameters so that it seems that it is important to adapt the method to each of your inputs. There's no universal recipe that will always work. You can try Persuade from this link, and I really want to thank you for your attention. Also all my lab mates, and especially Tony, and all the funding agencies. I hope that you enjoy the rest of the talks. Thanks.
So congratulations, Miguel, for your work. Very, very well explained and very clear. There seems to be a question here. Let me check and show on stage. So Sarah is asking if you are working in a species where structural variants aren't well characterized. How do you know whether a call is a true positive or a false positive? So in other words, how would you have a source of ground truth to measure accuracy?
Yeah, that's a very good question. Thanks.
So the data that I showed in this talk was on simulations. So we basically simulate variants, and then we test the accuracy, so we know exactly all the variants that we have. And we also tested Persuade on let's say real variants, like these ground truths. What we did was try to find samples where we would have both long reads and short reads. And since with the long reads are this technology that works better for finding structural variants, we define variants from the long reads, and then we test how does Persuade work on the short reads. So, and then this way is kind of an inference of these column accuracy. And then for we only have these defined sets of variants, of known variants in humans, and there we have like this kind of ground truth for sure. But like it's a mix of different things. Thanks.
Mm-hmm. All right. Yeah, I agree with you. It's a very nice question. And following this question, have you found any of the species more difficult to work with?
Yeah. So the genome size is important for scalability. So because we are optimizing parameters, so for small genomes, it's something that adds a little to the computation time, something that is kind of handleable. But if you want to work with like human genomes or I don't know, like any animal genomes, it's harder because you need way more hours of computation. So this will limit, like this is going to be the main limitation, I think, of Persuade. And actually, since we work with fungal pathogens, we will design this app specifically for us. But I think that I mean, everyone knows the resources they have and the patients or the problems. So, and then of course we see that there are some samples where you get more or less accuracy, even within the same species. And of course, each sample has like different coverage. It's known that you need very high coverage to have good, well-resolved structural variants, especially in mammalian genomes, for example. These also influences. Yeah.
All right, super interesting. And why do you mix both like parameters from different species, and test it in all the different species? Yeah. What is the reason behind that?
Yeah. So when we started this, we thought that maybe there's just one set of parameters, let's say best practices, that may work for everything. And it may be, we are using some default parameters. It may be that these default parameters are not, they are just the ones that other people use, or even that we thought they made sense. But maybe through this optimization, we would find something that was universal. Mm-hmm. So what we did was to test this pipeline on like how, when I find parameters that work on one species, how do they work on the others to test this hypothesis. And it turned out that in general, it's not the case. So each species seems to have like specific.
What about species that could be evolutionary closed?
Yeah, so I guess I like intuitively I expect them to be transferable, but we have not tested these because, I mean, for example, in the species that I showed you, for example, there are two Candida species. They are closer than the rest of them, but they are not, I mean, they're like a sponge and a fish. I tested it, like that you get an idea in vertebrates. So, and these guys, the parameters of one do not really work well on the others necessarily. So I think that I would not trust, I mean, if I was working on a close species to another that someone else has tested, I would still run the optimization. And the Persuade allows you to do this automatically, so it's no extra effort. Like it's just the same line of code, let's say, one single line of code. So that's what we did. Right?
That's super nice. So I don't see more questions in the panel, and although I have more questions, I'm gonna continue with the next speaker. But thank you so much, and congratulations again.
You can get in touch via email or the chat, whatever.
Sure, let's talk about it.
Thanks for the opportunity as well.
All right. Good, very good. So, all right. So we've got a strong representation from the Barcelona Supercomputing Center, since our next speaker is a PhD student there too. And let me introduce to Roc Ferriol Duran and his talk, which will be talking about COVID-19 and neutralizing antibodies. Welcome, Rock, and I'm going to play your presentation right now. The stage is yours.
Computing Center. But what I'm presenting today is a collaboration between the Cancer Immunogenomics Group at the Josep Carreras Leukemia Research Institute and the Pulmonology Unit at the Hospital Clinic. This project is entitled Neutralizing Antibodies as a Therapeutic Avenue for COVID-19. Before I start, let me thank the organizing committee for organizing this symposium and for allowing me to talk to you all today. So, why are neutralizing antibodies important for COVID-19? First, they are the natural protective immune response against this type of viruses, and their function, what they do, is to prevent the cell entry of these viruses. So they prevent the entry on the cell, but they also prevent the infection of neighboring cells of already infected cells. So they are there to prevent the reinfection of neighborhood cells. And how do they do this? For the case of SARS-CoV-2, so these neutralizing antibodies prevent the interaction between the receptor binding domain of the spike protein and its cellular receptor, the AC2. Then, since COVID-19 can be understood as a multi-organic disease that not only affects the lungs but affects also many other locations in the body, as it was shown in this paper, neutralizing antibodies can migrate to all these secondary locations, not only the lungs but also the other locations, and prevent the cellular entry there. Therefore, they can be an ubiquitous option for treatment that can not only treat the lungs but also treat other infection sites. So, in order to explore these neutralizing antibodies as a treatment avenue for COVID-19, what was proposed to us was to design B-cell epitopes so that could be targeted by neutralizing antibodies on SARS-CoV-2 proteins. Therefore, these epitopes from SARS-CoV-2 proteins could be recognized by neutralizing antibodies and be used as baits to isolate these antibodies and later produce them for treatment of COVID-19 patients. So, in order to design these epitopes, the first question that we asked ourselves was, what makes an epitope? What makes a B-cell epitope suitable for neutralizing antibody recognition? So first, neutralizing antibodies neutralize proteins by binding them on the surface of the virus. Therefore, we want to focus on epitopes that are on the surface of the virus, on the viral surface. Secondly, since proteins like spike are heavily glycosylated and glycans can prevent the antibody recognition, we want to focus on epitopes that are on the uncovered sites of the protein. Therefore, we will discard those epitopes that have glycans on top. Finally, epitopes can be at two, let's say, dispositions within the parental protein structure. Some of them can be exposed on the surface of the protein, and some others can be internal or buried on the structure of the protein. Therefore, here what we propose is that neutralizing antibodies will only recognize those epitopes that are exposed, and therefore we will discard those epitopes that are buried on the structures of the proteins. So with all these three sets of criteria, now, well, the next question was, how do we integrate and prioritize epitope predictions using these criteria? And also, how can we automatize it into a work package that can help us implementing this protein-wide? And this work package, we have called it BrewEpitopes, that stands for Brewing Epitopes. So as an example of our pipeline, for example, here we started from 4 proteins, S, N, E, and M. And on these proteins, we performed linear epitope predictions using 2 softwares, BabyPred 2.0 and ABCpred. And then we performed additionally structural epitope predictions using Discotope 2.0, sorry. And then we implemented our criteria. So we used a tool called EpiLocator to select those epitopes located on the surface of the virus. Then we used 2 glycosylation predictors, net N-glycan and net O-glycan, that coupled with LC-MS data enabled us to discover which positions of the proteins were glycosylated, and we used then epiglycan to discard those epitopes that have glycosylated positions. Then finally, we use, in terms of accessibility analysis, we use NetSurfP to predict which epitopes are exposed or buried, and then we use Chimera to visualize this exposition or the ones that are buried. Then to filter these candidates, we use Episurf, that basically restricts our analysis to those that are exposed. So with this, we reach a final selection of epitopes. Nowadays, we are working on making this protein-wide. So results. So how do we predict epitopes from these proteins? So let's focus on spike first. We obtain 51 candidate epitopes using linear and structural predictors, 44 and 7 concretely. And then we restrict to those that are exposed on the viral surface, that are 49, since the spike is a very extra viral protein, let's say. And then, as we saw, it's heavily glycosylated, so that's why we get to 36 when we select for the non-glycosylated epitopes. And then when we select for the accessibility, we see how 35 of them are on the accessible side of the protein. Then differently for nucleoprotein, since nucleoprotein is an intraviral protein, when we reach the filter of viral surface, we see how the candidates reach 0. And then for E and M, we see how the numbers again follow a nice, let's say, decrease, and we reach a final decent number so that these epitopes can be tested. So let's now move to the wet lab part, to the validation part. So the epitopes that we saw in the previous table, now they are synthesized in the form of linear peptides, and then these peptides are bound to beads, and then these beads are exposed to the serum of COVID-19 patients, where if we see a fluorimetric signal, it means that neutralizing antibodies there are recognizing our candidates. Therefore, we can compare whether there is recognition or not using this system against our epitopes. So, an example of this recognition, you see on the y-axis the signal, so the fluorimetric signal, and then you see different dilutions on the x-axis. The interesting comparison here is the comparison between the blue, the pre-pandemia patients that haven't seen SARS-CoV-2, and positive COVID-19 patients. So, as you see in this plot, every dot is a patient, and you see already on the left that is a set of or an epitope for which we didn't see great differences between pre-pandemia and positive COVID-19 patients. However, you see as every dot is a patient, you see how several patients already have higher signals of recognition than pre-pandemia patients. Differently and interestingly, in the right plot, you see how the difference here gets much higher, and you see how there are a good group of candidates that really have higher values of recognition of our epitopes, and therefore this peptide H, this candidate, would be validated and would be a good candidate to be used as bait for neutralizing antibodies. So, as I said, we want to focus on these patients that have greater recognition on the serum of these patients, and from the serum of these patients, we want to identify the most reactive patients, as I said. Then we want to identify the reactive antibody there. We want to isolate this and produce it for monoclonal antibodies, and then use them as treatment. So, with this, I have reached my conclusions. So first, we have developed a pipeline that builds on structural and linear epitope predictions and adds a set of prioritization steps that are based on the features that determine the recognition of an epitope by neutralizing antibodies. Secondly, we have built all this pipeline into a set of bioinformatic tools that integrate and prioritize epitope predictions, and importantly, they are not meant exclusively for SARS-CoV-2, but they could be implemented in other patients, in other organisms, pathogens. And finally, our preliminary experimental results indicate that some of our candidate epitopes are truly recognized by neutralizing antibodies in COVID-19 patient serum, and therefore they are useful as baits to isolate neutralizing antibodies. So with this, let me thank our collaborators at Hospital Clinic, Rubén López, Antoni Torres, and Laia Fernández, and also my supervisors at the BSc, Eduard Porta and Miguel Vázquez. Thank you all, and I'll be happy now to take questions.
So, well, I must say that a lot of work and very clearly shown. Very, very nice work. Congratulations. Now,
Thanks.
well, let's hope that, let's wait for some questions to appear in the panel. And the more I was listening, the more questions were coming up. But I think that the first ones that I took note was, what about these, all the famous SARS-CoV-2 variants? Do they matter here? Would it change depending on what virus kind of infected,
Yeah, thanks, Javier, and thanks to the committee again. It's nice that I get this question because it's an analysis that we are doing right now, but I didn't show it. So yeah, indeed. So what we did, we retrieved the variants that these alpha, beta, delta variants accumulate. And then what we try to do is to rerun the pipeline using this new FASTA sequence, let's say, with the variants, and see if our final epitopes are affected. But what I can tell you right now is that since let's say the region from the protein that these epitopes cover is quite big regarding spike. And since it's quite big, some of the epitopes might be affected by the variants, but it would mean that still there is a space for antibody recognition. That would mean that the variants might decrease a bit the antibody recognition, but it's not probable, let's say, that they will just lose them all.
Yeah, I see. So yeah, definitely you guys are really onto it. So very cool. And would it be useful to use vaccine-derived epitopes to the ones that you predicted?
I think some people tried, but it was harder than these RNA-based vaccines that are already there. So it would make sense. It would depend on the vector, so the type of vector that you use to expose these epitopes. But yeah, in my mind, it would make sense to use it as in a vaccine.
Okay. And moving to the bioinformatics pipeline, the BioPicTop, how do you assemble all the building blocks, all the tools? What do you use?
Yeah, so it's not containerized yet. What I'm using, it's just basically R language, and I'm planning on making a master script on Bash. But yeah, basically right now so I get data from different servers, and the task was how to integrate it, because sometimes epitopes from different tools were overlapping, and then you had to just put them together. But in sense of like containerization and that, it's not yet there.
Okay.
But I would probably go for Nextflow or one of these.
All right. And in terms for anyone interested in this type of pipelines, would you point out or highlight a specific step that was perhaps more difficult to you to set up?
So yeah, sometimes alignments that are really dispersonalized, because this is not using BLAST and aligning to something else. You get different results from different tools, and then out of the same sequence, let's say. And then this alignment, how to script it, was a bit of a pain, let's say. So that would be my answer.
Okay, we'll take note of that warning. Okay, so we need to move to the next speaker. I don't see there are more questions, so thanks again for your talk. Very nice.
Yeah, thanks.
All right, so let's move to our last speaker of this session. We are now very glad to listen to David Martinez, a master student at the SIMUS and Universidad de Santiago de Compostela. So we are moving to Galicia. And his work is about RNA editing and aging. So hi, David, welcome. And I'm going to share now your video.
Thank you.
So it is right here. Yeah, the stage is yours.
David Martinez, master's student in bioinformatics. I am glad to present my work under the title Dynamic A2I RNA Editing Landscapes During Aging Process. Aging is the most important risk for numerous human diseases, including cancer, cardiovascular disorders, and neurodegenerative processes, and is associated with the functional decline of all tissues. In recent decades, the aging of the population associated with the improvement in the quality of life, especially in the richest countries, has led to the study of the aging process being a priority to palliate both economic and social problems. A pioneering study by López Otín set the hallmarks of this process, whose study can lead us to know more about this process and find treatment to increase the health span, that is, the portion of life in which we are healthy. Aging is widely studied in the most prestigious journals. Traditionally, many studies have been conducted on DNA or genome, but in recent years, a new level of regulation has been discovered at the RNA transcriptome level. Recent advantages in sequencing technology have facilitated the reconstruction of the entire transcriptome by deep RNA sequencing, RNA-seq. This has made possible to easily study the enormous importance of the regulation of the transcriptome in the organism, both for the control of homeostasis and in problems derived from its deregulation. RNA editing is an essential post-transcriptional process that is able to modify transcript through insertion, deletion, and base substitution, provoking a chemical conversion of adenosine residues to inosine residues. We are using A2I RNA editing to establish a signature and see how it affects and controls the aging process. Unlike other aging hallmarks such as DNA or histone modification, changes in the epitranscriptome remains largely unexplored. Adenosine to inosine RNA editing is the most abundant RNA modification in mammalian cells and is catalyzed by the ADAR family of proteins. One of the main roles of A2I RNA editing is to modulate the innate immune response and preserve cell homeostasis. It contributes to increase the transcriptome complexity, expanding the repertoire of coding and non-coding RNAs with profound functional consequences. It participates in function as important as the stabilization of RNA splicing and silencing. An example of the enormous and specific capacity of this change in cellular control is in the case of GluR2 receptor, which is permeable when it is not edited and impermeable when it is. Certain fields in the editing pattern are related to cancer, atherosclerosis, and neurological diseases. Its deregulation has been associated with the different human diseases, including autoimmune and inflammatory pathologies, neurodegenerative and psychiatric disorders, and tumor. In recent years, sequencing technology through RNA-seq has allowed us to carry out expensive studies on post-transcriptional changes such as RNA editing. We decided to use ready tools. This script set allows analysis and filtering of editing sites, including A2I editing changes. For this analysis, we applied some scripts found in ReadyTools project. Especially, we've used ReadyTools known to explore known editing events in RNA-seq data we analyze, and ReadyTools de novo, which predicts de novo edited sites. These scripts carried out a series of internal filters to verify that the results are truly A2I edited sites. This filtering process has to discriminate real A2I edited sites from genome-encoded single nucleotide polymorphisms, technical artifacts derived of read mapping errors and read ends, because their quality is lower due to the sequencing process. In our project, multiple datasets have been used, including what come from our labs, IPS, and young and old fibroblasts. Raw data were initially inspected using FastQC, and reads were trimmed using Trimomatic. We removed adapter sequence and the last nucleotide. Then, high-quality reads were aligned onto the mice genome GRCM38 using BAM format. This process was accomplished by STAR using this aligner because it allows to find more edited sites. The use of a different aligner software causes that we find a different number of edited sites. After that, we execute a duplicate removal using Picard tools, and finally, the modified files are processed with ready tools. We obtain TXT tab format files, which undergo a filtering process to make sure that the site that we have found are truly edited. As we have multiple datasets to study, for now I have only performed the analysis of sample obtained from public MySkin datasets through known analysis. There are two main groups, young individuals 3 months and old individuals 30 months. We seek to compare young and old editing patterns to know how aging affects to this process and how this process can provoke changes during aging. All outputs are merged in one unique file to realize the filtering process. In our first analysis, we use a list of known edited sites obtained from ReadyPortal mixed with another one from Darknet, 107,093. All sites from all samples are joined and duplicates must be removed to obtain unique edited sites per chromosome position. Subsequently, we apply two filters. The edited sites must be present in 2 or more samples, in this case 5 total samples, and must have an edited percentage greater than 10%. Following the previous pipeline, we found that the editing profile is sufficient to segregate samples according to their biological age, as can be seen in Figure A, a principal component representation. Moreover, focusing only on misregulated A2I editing sites, that is, the sites that present a difference of more than 10% between young and old individuals, we observe a higher degree of similarity among samples, as can be seen in the Figure B. There are multiple sites that increase, particularly in the case of older individuals. Samples can be clustered by age groups using a heat map representation. Deburring in detail, we obtain the results shown in Figure A and Table B. There are 899 total edited sites located in 413 different genes. Our results suggest that A2I editing sites are mainly located in coding regions of RNA. Figure C shows the results of misregulated sites, that is, those with a difference of more than 10% between young and old individuals. We found 347 misregulated sites in 254 genes. There are a greater number of sites that increase their level during aging, and the same gene can have several edited sites. As expected, the RNA editing process increases with age, as can be seen in the Figure A in both coding and non-coding. We observe that only a small part of the total sites, 347 of 899, are misregulated. That's it. They present a difference of more than 10% between young and old individuals. Importantly, ontologies of genes where editing is reduced are associated to different steps of RNA life cycle. Editing sites can both increase and decrease their levels during aging, with edited sites appearing or disappearing in all individuals in both protein coding and non-coding genes, depicted in Figures A and B, respectively. These sites could be critical in the aging process and responsible for the editing contribution to aging. Finally, consistent with the literature, A2I editing process doesn't correlate with gene expression during aging, both protein coding and non-coding RNAs, as we can see in the Figures A and B. As a conclusion, we can say that ReadyTools is a highly flexible, reliable, and versatile tool for the analysis of A2I RNA editing. A2I editing profiles can segregate samples according to their biological age. RNA editing levels are altered with aging both in coding and non-coding RNAs, and there is no correlation between transcript abundance and A2I editing with aging. Thank you for your attention, and I will be pleased to answer your questions.
So, okay, thank you very much, David. It was, again, very, very clear presentation and very nice work from you.
Thank you.
Let's check now the Q&A panel and see if we got, I think, one question. No, this is from the previous one. No, it's for now. Okay, I think. So let's. I don't know if this was from the previous one. No, I don't think so. I'm a bit confused about this question because I didn't mark it as red, so
I think it's for me, but I don't know.
Sorry. Yeah, it's for this. I made it.
oh, thanks so much for the clarification. I was a bit confused. So okay, it says how important is it to remove duplicate reads for the analysis? For example, is it possible that you remove reads related to short transcripts, which may appear as a duplicates because they span the whole transcript?
Thank you for the question. I think that you can be right, but in this case, I'm looking for a unique edited site, and yeah, it could be interesting to do it again, the whole process, and obtain only the short transcripts to study them, if to know if they are more important. But now we are looking for the main edited site and the most important editing sites that affect some genes, some specific genes. I have not talked about it, but in this case, we are looking for edited sites located in especially coding regions and related to the inflammasome and NFKPV, some process of the inflammation, the inflammation provoked by the agent. So we are looking for that kind of genes, but it's good. It's a good idea. Thank you.
So, well, Miguel, if you want to expand your question or something.
No. I mean,
Yeah.
I was not doubting the analysis. I think this is like something that I asked myself. For example, for scan calling. And in variant calling, I've seen some papers that evaluate the effect of read duplication. It doesn't really affect much. It's like the last time I checked.
Okay.
But I was wondering on RNA-seq, which I'm not an expert on or anything. So it was just curiosity. Thanks.
All right.
Thanks a lot.
Thank you.
Thanks for the question and for the answer as well. Okay, let me check here. By the moment, there is no new question. But well, since I work also in the field of cancer, and sometimes we are detecting somatic variants and try to distinguish between germline variants and somatic variants, it came to my mind, like, you're using whole genome to try to avoid sites that are false positive. So what if whole genome is not available? Could you do this with only RNA-seq data?
Yeah, I think that I have worked only with transcriptome, but I don't need the genome. I mean, I'm working with the transcriptome.
Okay.
Only with RNA-seq data.
Okay, so, but yeah, it's just because you mentioned whole genome at some point in your presentation.
Oh.
And sometimes it's a bit, or perhaps I misunderstood, but I understood that. Okay.
Sorry.
And regarding your the tool that you of your choice, the ready tools, what are it's just a curiosity, what are the computational requirements to perform this analysis using this tool?
Do you mean in the computation power of my computers, for example?
Yeah.
Not very much. For example, for the whole pipeline, I need a star. That the star needs a lot of RAM memory, especially when I work with human RNA-seq. But in this case, I work only with RNA-seq from mice, so 60 gigabytes from RAM, and the processor and the CPU is quite normal. But yeah, I need. We have quite good computers in our lab, but not. We don't need a cluster or something like that.
Okay. And also regarding the choice of this tool, which seems kind of very suitable to run the analysis, is it? I mean, did you choose based on any benchmark, or is something that there are not many tools about this?
No, we didn't look for a suitable tool. We want most used in the field, and we want to look for a tool that gives us the best results. And we decide to use Star and ReadyTools because of that.
All right. And are you seeking for new data sets that you can explain?
Of course, in this case, I have only said that I have used a public dataset, but I'm using a lot of more, and and I'm using from our lab. We work with fibroblasts. In this case, we have used skin samples because they are quite similar, but skin samples have another type of tissues and cells. But in our case, we are looking for more public data sets from fibroblast samples, our samples, and maybe we are going to try to compare and study the obesity and standard diet comparison.
All right. Well, so we are getting to the end, and I cannot see any more questions. But remember that you can contact the speakers to the chat and private chats as well. So thank you very much, David, again.
Thank you.
And now also I want to kind of give a big applause to the 6 of you that made a great presentation, super clearly and very high quality work. And also, now we are going to take a 30 minutes break and be back at 4 to listen to our second keynote speaker, which is Laura Furlong. And this will be moderated by our colleague, Ines Rivero. And also, I'm going now to also remind you that now we will be sending the links to vote the best talk and the best poster presentation. So you will see that in the chat section. And also, I want to remind you that it's also time to use Needle app and do the quiz and try to win 30 euros in Amazon as a prize. So thank you, all of you, for listening, and see you in 30 minutes, guys. Thank you very much. I will end up now the session.